Queen Mary University of London, and my group kind of very much focuses on trying to use phenotypic data, like both clinical and model organism, to better understand rare diseases, both in terms of diagnostics, trying to improve the diagnostic yield, as well as discovery of new disease gene associations. So just as like very high level sort of view of what we're trying to achieve here for those of you who don't do sort of sequence analysis. So when we take a rare disease patient and we sequence them, there's like some 6 to 8 million variants in their whole genome sequence. And typically we can use like public genomic data such as Nomad, yeah, just to look for the really rare variants because we know it's meant to be a very rare variant. And we can look at like alpha missense and Revel and other tools to predict how pathogenic the variants are. But this kind of rarely gets you down to like a single causative variant. So for these Mendelian diseases, we're obviously looking for like a single causative variant or a pair of compound heterozygote variants in the gene. So generally what my group and others have done is to try and make use of the patient's phenotypes and to be able to automatically compare them to clinical phenotypic data from OMIM, say, as well as all the wonderful model organism data that you guys curate. And you'll sort of see towards the end of the talk, we're trying to bring in other sort of types of data, other omics, such as transcriptomics, to further help in this sort of filtering to help us kind of get down to the diagnosis. Oh, no, I meant to say you probably won't notice, but this is like spelt the American way because this slide's from my colleagues in the Monarch Initiative. So this is like a large NIH funded initiative. What we hope is going to carry on being NIH funded, where we all kind of come together to like work on these issues. And again, I'm probably preaching to the converted here, like why we need model organisms, but just to kind of give you some of the numbers. So when we're sequencing a patient, if we find a variant in one of the say 4 to 5,000 genes that we know are associated with human Mendelian disease from OMIM, then we can kind of work out, oh, well, that looks like a good candidate for that particular patient's phenotypes. But if we find a variant in the other kind of 75 to 80% of genes where the gene's not currently associated in OMIM with a particular Mendelian disease, then we're kind of stuck, even if it's a really kind of compelling looking variant. And that's where we can use the other knowledge from model organisms to fill in the gaps. So yeah, for mouse, fish, fly, worm, yeast, like we can kind of really increase this coverage to like more than 82%. And this slide's quite a few years old now, so I suspect this coverage is getting up over 90% nowadays. And again, just to sort of, you guys all know that obviously if you mutate the same gene in different species, you quite often get similar phenotypes. So, but the sort of challenge we're trying to fix is like to sort of automate this. So the fact that yeah, in Drosophila the eye is completely absent when you get these PAX6 mutations, where in humans you've maybe got an opaque lens, or in mice the eye is decreased in size. These are all sort of similar but related phenotypes. And this is where, like, all the way back in 2009, like the precursors to the Monarch Initiative kind of started to develop, being able to map between ontologies, such as the human phenotype ontology and the mammalian phenotype ontology in a sort of systematic way. So we can automate this process. And I can kind of go into more detail at the questions phase if people are interested, but to sort of keep it sort of fairly simple, what we sort of do in Monarch is we can take a patient's phenotypes represented here in the middle, and these will all be encoded using the human phenotype ontology that probably many of you have come across. And then we can automatically, using our kind of semantic ontology algorithms, compute how similar this patient is to a particular human disease, where we've got the curated annotations to a particular OMIM disease using the HPO. And we can do this for every disease in OMIM. So we can like rank the diseases. We can say what's the most similar one. What we can give it a percentage similarity score. And then if we find a high similarity between a patient's phenotypes in this particular disease, and we know which gene's involved, and we got a variant in the same gene in the patient, that becomes like an obvious diagnosis. But like I say, we want to increase our coverage. So we do things like mapping across to the mouse data. And we also make a lot of use of zebrafish data and elsewhere in Monarch I should say that they're kind of making use of all the different model organisms, but in our exomiser tool, we're currently using mouse and zebrafish. So we can do the same sort of automated comparison. So we can compare a patient's phenotypes encoded using human phenotype ontology terms against like all the mouse models in MGI, for instance, and work out what the most similar mouse model is. And again, there's obviously going to be a gene that's been disrupted in that mouse. And yeah, if this we got a variant in the same orthologous sort of gene in the patients, then that becomes a good candidate. So that's the basis of our approach that we kind of do over and over in our group. And we sort of do a lot of this in my team in the context of the IMPC, which I'm sure like the mouse, at least the mouse members of AGR are very aware of, and probably lots of the other groups. So the goal of the IPC is obviously to try and systematically characterize every single protein coding gene in the mouse, or at least the ones with a good one-to-one ortholog of humans. And to do this in a very sort of standardized way, so using the same genetic B6N background, to use standardized phenotyping screens on all the different lines. So there's some like 14, I think it's about 14 different mass production phenotyping centers spread out across the world that perform these assays. And I'm involved with the EBI and MRC Harwell in the UK of kind of collecting all the data and analyzing it statistically, trying to establish really robustly what phenotypes each one of these genes is associated with. And you won't be able to see the detail of this slide, but just to sort of give a sort of overview of the pipeline. So what we do in the IMPC is we try and phenotype 7 males and 7 females. It's like the sort of minimum number we kind of need to get a robust statistical signal without spending too much money per mouse or per gene that we're trying to assay. And obviously we try and do the homozygous knockout first of all, but as you know, for one third of those, it'll be lethal or subviable. So for those, like the heterozygotes, will hopefully go into the adult phenotyping pipeline that's run between 4 and 16 weeks. And then there's various assays. So I think the dark blue are the ones that are run in all centers. I always forget this. The light blue are just done in some of the centers, and the white ones are under development at the time this slide was made. But for all those ones that are lethal, there's also a complementary embryonic phenotyping pipeline that's run on the homozygotes. So at 3 different time points, varying, yeah, depending on when the lethality occurs. There's like micro CT, gross morphology, what else? High resolution electron microscopy and histopathology performed. So we kind of collect all that data. And the idea of the IMPC, so rather than doing like very, say, detailed phenotyping in one particular organ like the brain, they sort of try and capture across all the body systems as much as possible. So like there's more than 250 parameters measured potentially for each gene. So there's all sorts of neurological assays, metabolic, cardiovascular, lung, sensory, musculoskeletal, immune, reproduction, and then obviously that embryonic phenotyping assay. So hopefully there's a good chance that we'll cover at least one of the phenotypes you should see in the Mendelian disease when we produce the IMPC mouse. And my team in particular were involved in building, yeah, that automated pipeline based on that Monarch technology to sort of map between all the Mendelian disease associations we know about and all the IMPC mice we were producing to see if we got any good new mouse models or if we got any sort of suggested candidate disease genes. So we published this back in 2017 in Nature Genetics based on the first 3,000 sort of knockouts the IMPC had done. Over the years, sort of like people in my team, such as Pila Cachero, have kind of produced updates of what we discovered. So this is very much, and we do the same thing for the MGI sort of literature curated mice as well. So we kind of put all that together in the portal. And the idea is by putting both together, we kind of get really nice coverage and hopefully maximize the use of the mouse models for human disease diagnostics and discovery. So back in 2017, that original paper, there was only 316 kind of Mendelian disease gene associations where we had the IMPC knockout for the ortholog. And for the majority of these, like 72%, there'd never been a previous mouse model produced because it was, yeah, the IMPC very much focused on the dark genome, like new disease genes where mouse hadn't been made originally. And so, yeah, I should say the 360, there was like some phenotypic overlap between the human and the IMPC mouse model. There was obviously more disease gene associations than that. And more recently, this has risen to over 2,000 models where it's kind of IMPC data supporting the Mendelian disease association. And you can go off and order those mice and do extra experiments on them. And the nice thing in the example of the original paper, we managed to show that you kind of get nice sort of models across all the different biological systems pretty much, from bone to cardiovascular to hearing to muscle, neurological, retina. Yeah, for instance, Bardet-Biedl syndrome, we kind of nicely see the abnormal retinal morphology in the IPC mouse, as you'd kind of expect. So I'm not going to sort of dwell on the IPC sort of modeling too much. I'll sort of move on to more of the sort of 100,000 genome studies we've done, but a more recent paper we published in Disease Models and Mechanisms, was it last year? Yeah. And we sort of like kind of advertised all this on IMPC models, but we also did an automated look at the literature to sort of see how useful the IMPC data and how useful the IMPC mice have been for like novel Mendelian disease gene discovery. So here we've kind of like trying to identify those new papers that sort of announce the discovery of a new disease gene. They're kind of fairly easy to spot from the titles and the journals they're published in. And then we were looking, was the IMPC data useful? So the actual phenotypes that have been called, or was the actual IMPC mouse what was useful? So they ordered the mouse and did their own phenotyping, or was it both? So for the new discoveries, it was kind of 50-50, either the data or the line or both were kind of useful. And then there was a whole series of papers that we spotted that kind of weren't announcing the new discovery, but like for 29 of them, they'd kind of ordered the IMPC mice and done some further experiments to sort of look into the disease mechanism, sort of proving some utility. Yeah, and then so we put all that data on massphenotype.org, yeah, the main IMPC portal, but at QMUL we try and kind of host some more detailed data on our disease models. So and the sort of data you can get down to here. So in the database, there's like 2,400 like human diseases, like from OMIM, where we've got the human phenotype ontology annotations. We've got an ortholog that's been phenotyped in the IMPC. And then for more than half of those, like 1,300, we've actually can show automatically using our algorithm that there's some sort of phenotypic match. So for instance, there's a model of early onset diabetes, like a Mendelian form of diabetes that's kind of involving PDX1 that we kind of show gets a really nice high score with our algorithm because it's got impaired glucose tolerance. And I guess the important thing is, so these were like new mice that the IMPC had produced, so they hadn't been a previous published paper that had been picked up by MGI curation. So just to sort of show how the resources complement each other. And as I'm sure you know, like all the IMPC data gets fed into MGI and kind of hosted on there. So it kind of, yeah, it makes it tricky to sort of work out which data is coming from the IMPC sometimes, but we kind of know how to do that when we do this type of analysis. And there's sort of a bit of heresy, so I'm involved in this other sort of consortium that's kind of quite similar to IMPC, apart from it's using human cellular data. So yeah, I can imagine it will be popular in this audience, but it's kind of it's got its uses. I guess you can sort of it's like at least it's like human data, which some people want, and you can do it sort of quite fast. So you're doing knockouts in human cells. I could say the disadvantage is nearly all the data so far has been very sort of transcriptomics based. So in terms of the sort of work I do, I don't find it quite as useful being able to. Yeah, it's not a real phenotype in my eye, the whole sort of transcriptomic data, but just to say that data starting to emerge on this portal, that'd be kind of interesting to integrate with the model organism data as we go along. And then just to sort of get onto more of the sort of human rare disease diagnostic work we do. So this is like the Exomiser framework that Carol was mentioning. So this is developed by Jules in my team and in collaboration with Peter Robinson, who I'm sure many of you know because he's still got his position at the JAX and now he's sort of moved more full-time to Berlin Institute of Health. So the way Exomiser works, for those of you who don't know, we have a patient, we perform whole exome or whole genome sequencing, we call the variants, and they're stored in a variant call format file, a VCF file, and we capture their phenotypes using the human phenotype ontology. And we've recently developed something called a phenopacket that kind of really tries to integrate all that data into a sort of standardized format as part of GA for GH. And then what Exomiser does, say on the left-hand side, it'll take all those variants and try and filter them down to like only the ones that are rare, predicted to be pathogenic, segregate within the family if you've sequenced some family members. So you get sort of between sort of 10 or several hundred candidate variants sometimes. But on the right hand side, we're taking those patient phenotypes and comparing them to like everything we know from human disease, from the model organism databases such as MGI, our IMPC data, and the zebrafish data we particularly make use of. And then and then if there's any genes that still don't have any phenotype data from combining all that, we kind of use a guilt by association methodology using protein-protein interactions. So the idea being if the gene directly interacts with a gene that's known to be associated with a particular disease or a particular mouse model, we might assume the phenotypes may be similar. And that all goes together to like put a score on each sort of gene for how phenotypically similar it is to the patient's phenotypes based on all this reference knowledge. And then we can combine all this in a machine learning model to give a final Exomiser score. So hopefully you kind of left with a single variant that's rare, it's really predicted pathogenic, it segregates in the family, and the patient's phenotypes match what we previously know about the gene, either from OMIM or from model organism data. So we were kind of one of the earliest sort of people to sort of take this phenotype-based approach for rare disease analysis, but there's like been lots and lots of other tools published over the years now. So we kind of reviewed these recently in human mutation of just updated the table here. And like some of them perform really well. Like it's quite hard to compare the benchmarking, I'd say, because everyone's like benchmarking on different data sets. It's like a big issue in the field. But one of the things we kind of find to really be useful for the actual like human geneticists is like the software needs to be kept up to date. So not many of the tools manage to do that. As you can imagine, these are academic open source tools. You're not really funded to keep your software up to date. It costs money. So there's only a handful of tools that really kind of keep up to date. And the other thing to be useful, obviously, needs to use human assembly build 38, and you need to be able to sort of deploy it locally and run it on the command line in a high throughput way, because most of the clinical data is sort of stored in these trusted, secure research environments. So you have to bring your software to the data and analyze it there. And then there's all sorts of other features that various of the tools have, such as non-coding or structural variant analysis. And then a few of them make use of the model organism data. So Exomiser, our tool, this AI Marvel tool that was published, I think that was it was last year in a new journal from New England Journal of Medicine. So that was kind of nice. It also used model organism data. And some of them also use natural language processing of the literature rather than taking the wonderful curated annotations. So there's obviously pros and cons there. So like we've been running since 2014 with Exomiser, so like more than 10 years now, kind of doing the same thing, but like gradually making it better, updating the data all the time. But with large language models, it's kind of blown the field wide open and we're kind of keeping an eye on it. So every so often we do an experiment and we try and use the latest large language models to sort of do pretty much what Exomiser does in terms of the phenotype similarity searches. And they're starting to get pretty good, these large language models. So they kind of Exomisers here in yellow, and you can see some of them are starting to prioritize the actual diagnosis as the top ranked candidate or in the top 3 or the top 10, a fair proportion of the time. So you can imagine in a year or two's time, they're probably going to overtake, yeah, our semantic similarity approach, and we're going to have this kind of change some of our underlying code in Xamizer, but that's good. It's progress. And there's some other like agent-based approaches that we've started looking at that are even more kind of promising. Yeah, we've had sort of various master's students looking at these. Yeah, one thing I should obviously make clear is that Xamize is completely open source. It has been since day one, so like anyone can take it and use it, including diagnostic labs, researchers, companies. Yeah, there's companies such as Congenica in the UK who just make use of it, and we don't get a penny from it, which might have been a mistake in hindsight, but it's kind of great that it's out there and used. And just an example of this sort of paper came out a few years ago from the Children's Hospital in Los Angeles that they'd like taken Xamizer. We didn't even know they'd downloaded it, but they were able to install it, use it without getting in touch with us, which is kind of great. And it shows we've documented it. And they sort of basically built a whole interpretation pipeline just using Dragon variant calling and Xamizer. And were able to diagnose 72% of their patients as the using Xamizer, and it was at the top ranked candidate, and like 93% were in the top six ranked candidates. And actually, it was nice because they had like four diagnoses that in a gene that wasn't in OMIM at the time, but it had some mass model evidence, I think, or it might have been protein-protein interaction evidence. And they were like pretty sure about it from the Xamizer evidence, but they couldn't like announce it as a diagnosis straight away. But then like confirming publications appeared shortly after. So they kind of found Xamizer use of model organism data useful in that way. And then to just sort of change tack to the 100,000 Genomes project. So I was involved in this. I've sort of seconded Genomics England, which is a kind of not-for-profit company set up by the UK government to deliver genomics in the UK. So I worked for them between 2016 and 2018 as Director of Genomic Interpretation. It's kind of helping to deliver this 100,000 Genomes project. And as you'd imagine, the whole goal was to take patients within our NHS healthcare system and to sequence 100,000 of them doing whole genome sequencing. So these are a mixture of patients either with rare inherited disease or their family members, or patients with cancer where the like normal tissue and tumor tissue would be sequenced. And obviously the main purpose of this was to kind of end that long diagnostic odyssey, especially for the rare disease patients to get a diagnosis, but like also to like drive research, so to put all the data in a research environment and to kickstart a UK genomics industry, which yeah, there's all sorts of, I sort of still live in Cambridge and there's all sorts of companies that have been set up doing genomics recently. And really to sort of drive genomic medicine in the UK. So that's project finished a few years ago now, and by the end of it, actually, well over 120,000 samples are sort of taken and sequenced, and most of them are actually sort of rare disease sort of genomes, either the patient of the disease or their family members. And then the results have all been like fed back now to the original recruiting clinician, and we kind of know whether that a diagnosis was achieved or not, so that information comes back into the system. And I mentioned this research environment, so any academic kind of can get access to this data in the research environment. I think companies still have to pay to kind of come in and use the data. Yeah, it just involves your university actually having sort of signed an agreement of Genomics England, but it's fairly painless nowadays. And then you kind of get access to all those patient genomes, the clinical data that's captured on those patients, so particularly the human phenotype ontology terms. You can run like command line tools or R Studio. You can bring in your own software to do analysis. And then there's a whole bunch of other data. So as well as the sort of genomes and the human phenotype ontology terms that were captured at recruitment, they've kind of brought in other electronic health record data. So particularly the hospital episode statistics. So yeah, for each of these patients, like what would actually happen in their hospital visits. So that's all in the system. Digital pathology and mortality data, and they're still working on trying to get that primary health record, from what we call our general practitioner, the primary health contact. So while I was there, I was kind of one of the main tasks I had was to sort of deliver this marker paper on the 100,000 genomes project. So this was like the first 5,000 genomes as part of the pilot. And I'm not going to go through the whole paper, but it was basically just to demonstrate when whole genome sequencing would work for rare disease and when it wouldn't, in preparation for setting up like a real genomic medicine service in our national healthcare service. And also to show that tools such as Exomizer are kind of effective at helping the clinicians find the diagnosis, as well as something called a panel-based system. So this is where you create a bunch of um, genes that you think are going to be associated with the patient's likely disease, and then you only look for variants in those particular genes. So it's another way of filtering down the results. And we really sort of demonstrated in the paper that that research environment was critical. It really kind of upped the diagnostic yield. So Genomics England has its main sort of, I'd say, simple pipeline, but it's really sort of trying to look for the obvious diagnosis. And then quite often you need to go on and have researchers really delving into the data to get a further diagnosis in patients that are missed. And then really to sort of demonstrate that whole genome sequencing kind of perform better than whole exome sequencing. And then just to show that you could really do this genome diagnostics and discovery in within the healthcare system. So I'm just going to concentrate on the phenotypic aspects. So one of the reasons I kind of went to Genomics England for a few years to do this was because they really systematically collected phenotypes on each of the recruited patients. And the way they did this was with this sort of kind of fairly simple form, but it was effective. So for each patient, they kind of captured what specific disease they suspected the patient have. So something, for instance, Alport syndrome, which is a renal disease. And then for each type of specific disease they recruited, so there was about 200 of these specific diseases that they were recruiting for. There was a whole set of human phenotype ontology terms, such as hematuria, where the clinician would have to say, yes, the patient's got it, no, it doesn't, or it's unknown. So there'd be like a questionnaire, and then they could obviously enter additional phenotypes if they thought the patient had additional phenotypes. So this sort of led to capture of like hundreds of thousands of positive as well as negative phenotype HPO terms for each patient. So like a median of 4 positive and a median of 4 negative per patient. So as you can imagine, this is like a dream for me. Like with Exomizer, we got the whole genome sequence, we got all these human phenotype ontology terms and like 35,000 different rare disease families by the end to analyze. And kind of what we showed in the actual papers, like Exomizer could automatically recall the diagnosis as the top ranked candidate for 77% of the final diagnosis. And orange is the positive predictive value. So that's just saying that 18% of the variants that you feed back to the clinicians end up being a true diagnosis. So there's a lot of patients that are still undiagnosed. That's why this sort of orange bar is a bit low. And as you go out to the, say, top three or the top five, you kind of get up to 88% automatically recalled. So it kind of really helps the kind of clinicians at the end kind of come up with the diagnosis. And this is this panel-based approach was kind of equally effective but complementary. So putting the two together in the Genomics England pipeline was the best idea, where you could automatically detect 92% of the diagnosis in just a handful of variants that the final diagnostic labs had to review. Yeah, and I should say, like where we missed a diagnosis, it was because it was like a non-coding variant. So we initially focused on only the coding region, or it might have had a low quality variant call in the VCF file, so we would have filtered it out automatically just to keep our precision high. And yeah, just to give you some of the early successes, like there's obviously a lot more now, so and kind of give you an idea how this works. So this is what one of the earliest patients diagnosed. So Jessica, she was age 4 at the time. So she had epilepsy, various developmental delay phenotypes, and all the standard genetic tests were negative. So she got sequenced. She had those sort of 6.4 million variants in her genome, but the parents were sequenced as well. So by looking at just the rare variants and just the ones that are coding, causing a change in the protein, and just the ones that were different to her parents because the parents obviously weren't affected by the disease, and then the ones that were in a sort of likely candidate gene. It kind of got down to this final diagnosis of this de novo deletion in this SLC2A1 gene. And that causes GLUT1 deficiency syndrome. And the nice thing was that you can like partially treat this by switching a diet to a sort of ketogenic low carb diet. And because it was a de novo variant, obviously the parents could go on and have other children and not have this worry that the sort of condition was going to recur. Yeah, and this is like it was like a lot of positive news when the paper finally came out, which is kind of nice to see because quite often you publish something on DNA sequencing or genomics, and you get a lot of tabloid press in the UK that sort of publish horror stories, but it was all very positive, like the news stories that we got when we released the paper. That was good to see. And there was a lot of sort of clinical utility that we demonstrated. So the average diagnostic odyssey for these patients was 6 years, and they'd gone to hospital 68 times for like specialist appointments. So it's like terrible for the patient and the family, but also terrible for our NHS. So this is all paid for by the taxpayers and UK government, and it's just for those, yeah, the first 5,000 patients, this had cost £87 million. And in terms of clinical utility, it's kind of early days when you first issue a diagnosis, but like 24% that had some immediate utility. Yeah, a lot of the time it just informed on future reproductive choices. If it was a de novo variant, there was like a low chance it was going to recur, obviously. And but there were some patients where you could change medication or enter them into clinical trials. And then in the UK, so I said the 100,000 Genomes project finished a few years ago now, but we now have our sort of genomic medicine service running. So like every patient in England, at least as part of NHS England, if they have a suspected rare disease and it's part of this registry for whole genome sequencing, they'll automatically get a whole genome sequence now, and it will get analysed by Genomics England, kind of using Exomiser, using that panel approach, and using like the model organism data that all of you guys have sort of painstakingly curated. So it's kind of a really nice use of the data. And there's going to be like 500,000 going for this genomic medicine service, as well as kind of all sorts of other genomics going on in the UK. So that was like the sort of the positive news story about sort of genomics in the UK. But actually, from the 100,000 Genomes project, our final diagnostic yield was around 20% of families where we kind of issued a molecular diagnosis. So we still got 80% that are unsolved, which is like terrible for the patients. But in terms of research, it was obviously that data is just sitting there in the research environment. We've got the whole genome sequence. Like if it is a monogenic genetic disease, the answer's in there somewhere, most likely, unless it's a really complex structural variant. So we just need to kind of carry on doing research, and there's obviously enough here to keep me going until I retire, probably. But this is our main focus in the group now, to really trying to address these unsolved cases. So looking at some low hanging fruit, so re-looking at the coding variants, the SMVs, and then there's like new disease genes being discovered all the time. So kind of going back in and looking at those, focusing more on the non-coding space, seeing if we can find splice intronic variants or regulatory like enhancer variants. That's way more challenging. And then structural variants which haven't been looked at so much today, but we're doing a lot of long read sequencing, which is going to help a lot with that. And then finally, novel disease gene discovery. And this is where the model organism data is probably going to help the most. Just to kind of give you an example of the sort of reinterpretation, going back and trying to look at the low hanging fruit. So we published this paper, Letitia and my team last year, where we kind of went back and looked at all the unsolved cases with Exomiser and took a very conservative, just looking for de novo or loss of function or known ClinVar pathogenic variants that hadn't been discussed by the labs before, and the ones that are showing a nice phenotypic similarity or in a newly discovered disease gene that was on the disease panel, and where we classified it as pathogenic by ACMG. And then we kind of reviewed all the evidence and basically got 463 new diagnoses. So, and you can keep repeating this every few months and get new diagnosis this way. So it's a really important activity. It's not like that interesting research wise, but like for the patients, it's very important to keep doing this reinterpretation. We've started looking at the non-coding, especially the cryptic splice variants. So, yeah, Peter Robinson, I mentioned earlier from Jax, like while he was at Jax, he developed the scrolls. Well, I should say he's still at Jax, but his Jax team actually developed the squirrels software to like look at cryptic splice variants, and then Illumina developed Splice AI. So we've run these tools using Exomizer across the intronic regions as well. You end up with tens of thousands of candidates. That's the drawback. So it's kind of hard to go further. But we've recently been working with groups who have done transcriptomics and proteomics on the same samples. So if you can show that the expression of the gene or the protein is like a complete outlier compared to like the wild type, and you've got an interesting variant and you've got a high splice AI or scroll score, then it becomes a diagnosis. And then finally, to sort of finish some of the novel disease gene discovery approaches where we can use the model organism or our network approaches and exomize the protein-protein interaction approaches. So one exercise I did was to look at those sort of 28,000 unsolved cases and try to see if there's any gene that was not a known disease gene, but where like a really high scoring exomizer candidate was coming up in three or more independent families. Now I'm looking at three or more because that's normally like the kind of baseline for like publishing a paper in the American Journal of Human Genetics or some other journal that kind of people will believe it's a new disease gene. So here the evidence is coming from mouse or zebrafish or from protein-protein interaction data. And so, yeah, if you have this strict looking for three or more independent families, this is rare disease, so it's kind of hard to find that. But we did find like 24 new disease genes that kind of look like they got pretty strong evidence using this criteria. And some of them have started to be published already. So this VPS4A that was involved with a neurodevelopmental disorder. And then some kind of individual examples. So like these are like individual cases. So we've not got the three or more cases, but just to give you a flavor of some of the things we can find for the patients. So this patient had intellectual disability as a sort of general syndrome, but also microcephaly, atrial septal defects, bone ossification, and short limbs. And we found, running exomize, that we had this kind of really highly scoring rare de novo variant that's predicted damaging by Revel and MVP, which are kind of, yeah, leading algorithms that we use for this task. So it causes a methionine to a valine change. And then when you look at the phenotypes, they're really sort of nicely matching a mouse mutant of the ortholog of this gene. So the mouse has microcephaly, has the atrial septal defects as well. And yeah, it doesn't have short stature, but it has short limbs and it has bone ossification sort of changes as well. So a similar sort of story with another patient. So we found a de novo variant in the gene not previously associated with human disease, this NCOA6. If you look in Nomad, this is a very haploinsufficient gene, so it kind of fits with the autosomal dominant model. And again, we had a mouse mutant that seemed to recapitulate some of the liver abnormalities. I think this one was an IMPC mouse where, yeah, it was homozygous lethal as you'd expect from the haploinsufficiency, but the hets had liver phenotypes, but not particularly specific phenotypes. So ideally, you'd kind of go and order the IMPC mouse to do some further assays. And then a final example with a sort of cleft lip phenotypic match where, yeah, again, the gene's not C-plane2 is not associated with human disease at the moment, but we found a nice de novo predicted damaging variant and the phenotypes match between the patient and the mouse. And Leticia, again, in my team, is sort of doing this more systematically. So she did a whole PhD on hearing loss. So she took all the IMPC novel hits for hearing loss and kind of screened the 100,000 genomes data to see if there's any evidence for any of those IMPC mice sort of candidates. And similarly, like starting with GWAS loci. And then just to finish off quickly, some of the work we've been doing recently on novel gene discovery, we've developed a gene burden testing So this is not initially using phenotypic data. We just running XMizer, and then we're looking for enrichment of particular classes of variant in genes in cases versus controls. So the cases will like be all the cases that we've recruited for hereditary ataxia, for instance, and the controls will be non-neurological cases. And when we do this, we've developed a statistical pipeline. You kind of uncover a bunch of known disease gene associations as well as some novel candidates, and so such as UCHL1 down here, that's just above the significance level in the FDR rate. And yeah, one of our early successes, we discovered this link between hereditary spastic paraplegia with this pipeline in UBAP1, and yeah, it was a super strong signal. The XMIT candidates this time were based on protein-protein interactions between a well-known spastic paraplegia gene, VPS37A, and this UBAP1. And yeah, we were very excited and kind of wanted to publish it straight away. And then the paper kind of came out almost the next month from another group, but we were kind of able to publish our findings. And we've published the findings earlier across the whole 100,000 genomes project, where we kind of ran this pipeline earlier this year in Nature, where we described some 69 candidates in the actual paper. We since working on the correction actually, because we discovered something silly that we've done in the FDR correction. So we'll be updating that hopefully soon, where we got like 11 candidates that we're way more confident on now. So including dilated cardiomyopathy and this new PSMB11 gene. And then as another way of using the IMPC data, in particular, Pilar Cachero and my team's published a bunch of nice studies recently. And the main basis of this is to take both MGI and IMPC sort of lethality data. And what we discover, we can sort of categorize all genes into either cellular lethal. So these are genes where you do like a human cellular assay and you discover the cells don't grow or they die if you knock out the gene, or developmentally lethal. So these are genes where the human cells, for instance, are okay, but in the mouse, you get lethality. And then you get subviable lines where you knock out the gene in mouse, and obviously the line's subviable, and then the viable ones where you either have a phenotype or no phenotype. So Pilar's developed this sort of spectrum of essentiality, these sort of 5 different categories. And what we find is these sort of mouse lethal genes that are not cellular lethal are particularly enriched for disease genes, particularly developmental disorders, as you'd imagine, and dominant disorders. And they, if you look at any of the genes in this category, they're a really rich source of new disease genes. So in the original paper, I think we published a table of our top 10 candidates, and actually like 3 of them have already been published as new disease genes since the paper came out in 2020. So that's kind of a nice promising approach, making use of the mass viability data particularly. I think with that, I can thank my team. This is like our last Christmas dinner, I think. And all the patients in particular from the 100,000 Genomes project. And as you can imagine, there's like almost 1,000 different clinicians and bioinformaticians that were involved in this project. My colleagues in the IPC and then the Monic initiative. So that's probably quite a long talk, but I can hopefully got a bit of time for questions.
I think so. Thank you so much, Damian, for that
Stop
fantastic
sharing a
talk.
second.
It's it's one of the reasons we do what we do is to help interpret the human genome so that it becomes a valuable tool for improving human health.
Yeah.
And so the examples you gave just really highlight the importance of what we do and its translational relevance. And so thank you for that. We do have time for a few questions. If people have one, if they want to raise your hand, I can unmute or you can put it in the chat. I had a couple of questions that I thought we'd start off with, which is a lot of what your work is based on currently is sort of that semantic mapping between HPO and MPO
Hmm.
terms, which I know is an ongoing effort. Are there still large gaps in those mappings? Are there still mappings yet to be defined and created? Are there,
Mhm.
what do you see as the major need for that
Yeah,
support?
and there's obviously people in your team that are kind of
Yes.
been involved in that, the
Yeah.
logical definition approach. And I don't think there's gaps in particular, but it's not optimized, I'd say. Like you can still see like places it can be improved. And there's people like Nico and Monarch that are spending a lot of time in Chris Mungle, like trying to improve those mappings. But like one interesting thing that's come up recently of the LLMs, which I think go into, is like this embedding approaches.
Yeah.
Yeah, where you just like for every ontology term, you make an embedding and then you sort of automatically calculate like the cosine angle between the vectors, and you just use that to map between ontologies. And what people are starting to show is this is actually working really well. It's like scary, but it's kind of working all pretty much as well as all the painstaking mappings we've been doing for a decade. So it's like, yeah, good and bad news. But
And
yeah, if it starts to get better, that's a lot easier to do.
right. And you talked a lot about the fact that a lot of your work so far uses zebrafish and mouse data. Do you have plans? Do you see a way to incorporate sort of phenotype and disease annotations from other model organisms into this work to maybe improve the accuracy or the scope of phenotypes covered?
Yeah, I mean, that's interesting because I think like one of the main things that sort of stopped me a bit in the past was that mapping, yeah, between HBO and because everyone's like using different ontologies. It's been the bane of our life, right, in this field. Like, how to do all these mappings, but maybe the embeddings, maybe this is actually that's going to be the positive benefit that we can just map everything to everything quite easily now. And we can
Right.
start show, I mean, and then one thing my team's worked on with others in Monarch is this fee-val evaluation framework, so we can really sort of rapidly evaluate whether things are improving or getting worse when we sort of change our knowledge graph in Monarch or our Exomizer database. So yeah, it might be a good time to try some of the other model organisms.
There's a question in the chat. Damian, awesome work. How do you prioritize which genes and phenotypes to pursue next in IMPC? Also, what are your thoughts on the scope for filling gaps within the existing gene phenotype matrix?
Yeah, that's an interesting question. Like, so us informaticians always try to like try and direct which genes were going to be like phenotypes next in IPC. I don't think we ever had much success. Like the production people always had their own ideas, and they were mostly right, I must say. Like this focusing on the dark genome is interesting anyway. And there's a lot of really interesting work that people I've seen people do where they kind of predict the phenotypes that are left anyway, and then you could use that to prioritize.
Great.
Yeah, I think Arjun's saying the embeddings work very well as well. Yeah,
Yes, yes,
I think everyone's finding that. But I mean, embeddings only work because of the fantastic definitions and the ontology terms, right? So
exactly.
everyone was like, "Oh, we didn't need all these ontology work," but yeah, you did
Yeah,
because
yeah,
only
exactly.
works because the ontologies exist.
Yeah. Other questions or comments for Damian?
Yeah, and there's a similar comment for all the people who think AI can just replace model work because yeah, half the AI is based off model organism data. So
Yeah, I mean, I think it's pretty remarkable what what you can do with it, but it does become kind of circular, right? You couldn't do what you're doing with it without the work that we did
yeah,
to do the structured knowledge representation.
yeah,
But that's okay. As long as it advances the field,
yeah, yeah,
it's a good outcome.
yeah. Initially, you get cross about these things, don't you? Then you realize it's good that you've advanced things.
All right. If nothing else, again, Damian, thank you very much. And maybe we can get you to come back and give us an update uh, in another year or so.
Yeah, I'd be very happy to.
Okay, great. Thanks so much. Thanks everybody for joining. Oh, Ann Quiddick has her hand raised. Ann,
Nope, I was trying to log out and hit the wrong button. But
you were
thank you
trying to
very much. That
applaud.
was an excellent talk.
All
Thank you.
right. All right. Thanks, everybody.
Thanks.
Okay, great to see you all.
Great to see you, Damian.
Yeah, thanks. Bye.
Bye.